camera and being recorded. And our first speaker is Camille Delavaux, who's going to be introduced by her advisor, Jim Beaver. So take it away, Jim.
You're muted, Jim.
Is it unmuting, Jim? No. Let's see. I was hearing you just a minute ago. I wonder what's going on. Oh, sorry.
Thanks, man.
It was my fault. That was totally my fault. I clicked the button where no one could do it. And he's like, why can't I unmute? Sorry, go for it. Sorry to waste time.
No worries. So I'm very happy to introduce Camille. Camille is a fifth-year graduate student working on various aspects of how microbes might influence plant biogeography at multiple scales. And today she's going to be talking about work that she's been doing locally on KU Field Station and other properties.
All right, thanks. I will try to share. All right, can everyone hear me and see the screen? Awesome. Well, yeah, I'm excited to share some of this work with you that I've been doing over the past 2 or 3 years, and some of it's well kind of along, and then some is kind of to get some feedback from you, and just sharing ongoing work also. And broadly, it's looking at the evolution of plant microbe interactions in post-agricultural grasslands. So these are disturbed grasslands that are just abandoned agriculture, basically. And so the first part that I'll talk to you about is the evolution of mycorrhizal response in post-agricultural grasslands. And so the title is Microbe Plant Interactions, and here it's specifically looking at mycorrhizal plant interactions. And what are these mycorrhizal plant interactions? So here I'll be talking about arbuscular mycorrhizal fungi, or AMF, which are the predominant kind of mycorrhizal fungi in tallgrass prairie ecosystems and are really important in late successional plants. And so this fungi actually colonizes plant roots. And like you see here, this is a root that's been colonized, and the fungus helps bring mostly nutrients to the plant, and in exchange, the plant provides carbon to the fungus so it can keep growing and keep supplying nutrients. And again, these are really have been shown to be very important in late successional plant species and tallgrass prairies. And what's pretty incredible about this AMF plant interaction is that it's been evolving. So these two partners have been co-evolving for at least 400 million years, where AMF have been thought to actually help plants like physically colonize land through kind of serving as a makeshift root system for aquatic plants. So this co-evolution has been ongoing in native sites. Oh, I lost my arrow. But this might be altered. This relationship might change due to anthropogenic or human-caused disturbance, especially with novel combinations of AMF and plants. And so here the question is broadly, how do these plant mycorrhizal interactions change with human disturbance? And specifically in post-agricultural sites here again. So for this project, we selected 4 plant species that are all native plant species here. And so we have dogbane, milkweed, ironweed, and goldenrod or solidago. And you'll notice these are all really early successional. And the reason that this is the case is because we wanted to choose plant species that occur both in native sites and also colonized post-agricultural sites. So those are the kinds of species that do that. And so we worked with 4 native plant populations of these 4 species and 4 post-agricultural plant populations of these species. And this is all in eastern Kansas, no more than like an hour from Lawrence. So it's pretty local. So we collected seeds of each of these 4 species at native and post-agricultural sites. All right, if you just want to focus on the green versus purple here for now. So we compared native plant populations in the green and post-agricultural plant populations again. And so we grew all of these 2 populations in sterile soil with no AMF or in with native AMFs that are actually from prairie systems. So presumably they've co-evolved with native plants. And then non-native AMF, which is from a culture collection. So they're really just not native to the area. Okay, and again, the big kind of point here is to see if there's a change in this plant AMF relationship from native to post-agricultural plant populations. So are they evolving and changing in this relationship when they recolonize post-agricultural sites? Okay, so one of the most basic things we can look at with looking at mycorrhizal fungi is the actual root colonization. Remember I told you they colonize root systems. And so here we find that there is a difference between the native and post-agricultural plant populations, where there's a significantly greater mycorrhizal colonization in post-agricultural plant populations. And this is regardless of what kind of inocula or what planting, they just have more internal root colonization. Okay, and so before you try to kind of understand this figure, if you could just focus on this initial first bar, you can see my arrow. So here we're looking at mycorrhizal response, which is basically the biomass, the improvement in biomass or growth of a plant due to having this, having AMF. So relative to the sterile control. So here, if it's above 1, then there's a benefit to having mycorrhizal fungi because it's making the plant grow bigger, basically. So what we find here is really cool. It's evidence for evolution of this mycorrhizal response because there's a interaction between the plant population type and AMF inocula. And so to put it in more understandable terms, native plant populations grow best with their native inocula. So they're showing kind of a preference for native inocula, while as the post-agricultural plant populations of the same plant species are growing best with non-native AMF inocula. Okay, and then if we go down into species, so each of these 4 species, and look at the differences in mycorrhizal response between the 2 populations. There are 2 plant species that show evidence of an evolution of this response. And the first is dogbane here that actually shows a reduced response in post-agricultural sites, which is pretty kind of what we expected because post-agricultural sites usually have a degraded mycorrhizal community, and so it makes sense that these post-agricultural plants are evolving a reduced dependence on mycorrhizal fungi. So that makes sense. And then ironweed is a bit of an outlier. We're still trying to figure this out. So if you have any ideas why this might be happening, but there's actually a significant increase in mycorrhizal response in the post-agricultural plant populations for this species compared to the native populations. So yeah, that's still in development. So why does this matter broadly? We find that there's evidence for really fast evolution of mycorrhizal response in this plant-mycorrhizal relationship in post-agricultural grasslands. And when I mean rapid, these lands have been abandoned from 10 to 50 years ago. So in that time span, this relationship has altered pretty significantly. And we can talk about this a little more, but from a restoration standpoint, I find it really interesting because we're looking at the same plant species in these two types of sites, and they're all native, and plant population, so if it's from a native site or a post-agricultural site, might be really important for restoration if you actually care about maintaining the native plant AMF relationship. Okay, so that was the relationship with a beneficial microbe, but another really important group of plant-associated microbes are root pathogens. And so the second part is more recent, but we set out to look at, identify first root associated pathogens from these same species. So I took roots from these same four species, from remnant, from native sites and post-agricultural sites, and grew them on different media to try to isolate any root associated fungi or oomycetes, which are known to be really important pathogens. So here we're just culturing all root associated fungi and oomycetes. And this is kind of an aside, but I've had a really long adventure in figuring out what the best culture storage methods are to be able to have kind of similar, like not as far apart generation times between isolates and different experiments. So I'm happy to share any of that or talk about that also. But this is really helpful in repeating experiments with the same or very similar strain. Sorry about that. So in this root pathogen section, there are two tests that we did this spring, which was a bit stressful, not knowing when we were if we were allowed to finish it or not, but we successfully completed them. So the first is identifying all these things we cultured, what is a pathogen? And so every inocula or every culture that was grown from a given species was then put on that species to see if it would reduce its biomass and act as a pathogen. And then the second test is kind of informed from the first test. What is the pathogen specificity? Or basically in this example, if we have a pathogen, something we've identified that could be a pathogen on dogbane, does it just impact dogbane, or is it kind of a universal issue for all of these early successional native species? So we ran 2 greenhouse experiments to test those. And these are super preliminary results, so I'd appreciate your feedback or thoughts. But I just wanted to define what a pathogen is clearly and kind of prime you for seeing the pretty busy graphs that are upcoming. But for us, a pathogen is something that when you add the inocular or the culture, it should reduce the biomass of the plant relative to a sterile control. And so this little bar here is kind of what you should expect to see to identify a pathogen in the next graph. So the growth of the plant is reduced, right? It's under the line compared to sterile. Okay, so we tested many different isolates. And this is the initial test. It has a pretty low replication because we did test so many. And we were able to test several on each of the different plant species. And I guess this is usually not an interactive situation, but does anyone see or like notice anything that's kind of weird in this graph? Oh, I guess everyone's muted. Well, we're expecting it, if it's a pathogen, to be under the line, right? And most of these are actually above. There are a few that are could be candidates for being pathogens to that plant species, but many of them are actually significantly, right, red and green, above the line. So it seems like they could be somehow benefiting the growth of the plant. All right, so from this, we took the most likely contenders that were either really positive or really negative from each plant species and tested them on every plant species to do the specificity test. And here we were able to replicate a lot more, and we got a lot more significant results, which is cool, and some negative ones, but it's still pretty overwhelmingly positive. So, but something important to note is that this is only above ground biomass, and I still have below ground and total to really look into more. So this is only thinking about above ground, like the shoot growth. And then something that I thought of was, even though these plants might be growing better or worse, I saw that there was maybe some differential survival, and there actually is. So some of these inoculums are disproportionately causing death of plants. So that's a pretty strong pathogen signature, but this is really initial also. So, but I just wanted to share. Okay, the third part is we cultured all these root-associated fungi and omycetes, and we're testing like one functional trait somewhat through these greenhouse tests, but we don't really know what they are. So we're hoping to do, we're planning to do Sanger sequencing to be able to actually hopefully identify some of these, most of these inoculums, and I'm hoping this can solve or maybe try to inform why we're seeing these weird patterns. But so that's ongoing. All right, I know I threw a lot at you, but finally, the last part is looking at the evolution of mycorrhizal induced pathogen resistance, which may occur this spring. That would be awesome. But so the idea is that AMF don't only help with nutrient uptake that you usually hear about, but they also help with lots of other non-nutritional benefits. And one of them that I've always been really excited about and interested in is pathogen resistance. So basically adding these fungi helps mitigate negative effects of pathogens. And so the hope is to actually test if there is a difference in this mycorrhizal induced resistance between the native and post-agricultural plant populations. Is there a change? And yeah, this is just another way to show it, where if we have this potential pathogen with no AMF, it will maybe hinder the plant growth, but then if you add AMF, it could kind of alleviate that negative effect of the pathogen. So I'm really excited about this, but it's kind of a long ways away, possibly. Yes, thanks for your time, and I hope that wasn't too much, but I just wanted to share the whole picture of kind of the sequence of projects. And thanks to you and lots of other individuals that helped, and organizations, and especially the Cave Field Station for helping fund this twice. So I'll take any questions or thoughts.
Excellent. So I've just made it where people can unmute, but people just send a chat and I'll call on people. So I think Hayan had a question that he wanted to ask first, so go ahead.
Yeah,
Hayan.
thank you. Yeah, so Camille, very interesting talk.
There. And Hayan Chow is going to introduce Saket, who's both from the aerospace engineering department. All yours.
Okay, yeah, thank you, Ben. So hello, everyone. My name is Hayan Chow. I'm a associate professor from aerospace engineering department. The reason why I'm here together with my student Saket is because we are using kind of collaborating with KBS researchers such as Sheena. We are trying to collaborate with Dr. Brian Foster as well, and Jude maybe later down the road. So I mean, kind of for our perspective, essentially our perspective is UAS, unmanned aircraft system-based remote sensing perspective. We're hoping we have an ongoing project funded by USDA. Saket will presenting the result, and then some other work we are working on right now, hopefully for some later real opportunities. Okay, so Saket is my PhD student. He's in his third year. His focus is essentially multispectral remote sensing unmanned aircraft system. So Saket, please, yeah.
Can you all see the presentation? Yeah, okay.
Yes.
So good afternoon. So today I'm going to talk about multispectral and thermal remote sensing using K-Hawk UAS. Okay. So I'll be discussing two projects. One is the wildland fire monitoring and mapping using UAS, NIR, and thermal imagery. The second is intercomparison of UAS and neon-derived NDVI for hayfield observation. So I'll start off with the FIREWORK. So this is a part of the USDA National Robotics Initiative that we're working on a collaborative autonomy and safety system for teamed human and UAS systems for wildfire environments. It's a collaborative effort between 3 universities. We have Dr. Hu from Georgia State, who's working on fire simulation model, Dr. Minshing from University of Missouri, who works on the control, and Dr. Chow's team. So we are in charge of the fire sensing and wind estimation using UAS. So our current objective is we want to develop fire sensing and wind estimation models using UAS and combine that data assimilation with Dr. Who's fire simulation model, which can enable driven wildfire simulation for future scenarios. So the other team members in this project are Dr. Tian, actually he graduated this year, and Harold Flanagan. And we have a couple of master students, that is Jackson and Justin. So we went out for a UAS fire flight mission last year in 2019. At the Anderson County Prairie Reserve on October 8, 2019. Yeah. So our main objectives here was to monitor the fire from UAS. We wanted to map the entire fire evolution from time t equal to 0 to t equal to t. We also wanted to estimate the rate of spread of fire in terms of the speed of propagation in each direction. So we had 3 aircraft flying. We had a K-Hawk 55 that you can see down there. We had a DJI quadcopter that was from the Missouri State University. They had come with their drone. And we also had a Zephyr 3 flight. And this flight was used for mapping the changes in crops due to burn. So the K-Hawk 55 UAS was developed for fire monitoring. We have the aircraft equipped with 2 cameras. One is a FLIR ViewPro R thermal camera, and the other one is a GoPro near-infrared camera. So we have these 2 cameras simultaneously taking pictures of the ground, and which we'll use for fire evolution, as you can see further. We also had a aero probe, 5-hole probe for wind estimation, so 3D wind estimation while flying as well. Additionally, we had a ground wind weather station, which is a Campbell Scientific for measuring wind and turbulence on the ground. So the burn was conducted on October 8th during the late morning, afternoon time. The temperature was about 73 degrees Fahrenheit with heavy winds from the south, about 13 miles per hour. So as you can see on the figure here, so there were 2 teams that were initiating the fire, starting in the start, probably in the, in the northern boundary somewhere there. One team moved towards the west, other 2 moved towards the east, and they met in the southern boundary, right where close to where the circle is. So the first sighting of the fire from the UAS was about 11:38 a.m. and we estimated the fire to last about 35 to 36 minutes. We had 2 flights. The one flight on the left bottom was doing loops on the northern boundary. This was just to estimate and map the fire propagation in the northern boundary. And we had to land the plane due to battery and take it off again for a flight 2, which is on the right, bottom figure. So we did multiple loops around the field with the intention that with each loop we can generate a map, and each map will give us a fire evolution at that time stamp. Both these flights were conducted at 120 meters with pre-programmed autonomous flight trajectory, and we had about 2 minutes per loop, that is in the bottom flight figure. So after the flight, so images from each loop are georeferenced and orthorectified to produce sequential maps. So our goal here, as I said, so for each loop, we can get one map, and from the map, we can extract the fire line. So in which case we have time sequential fire lines due to each loop. So we used Agisoft Photoscan professional software for orthorectification. The NIR images were extracted from a 1080p video at 30 Hertz frame rate, and thermal images were acquired at 1 Hertz frame rate. So I provided an example of our map. So on the left bottom, you see the near-infrared map. The circles are basically the flight locations, or rather it's the images we've used for this loop to generate this map. On the right, we have a thermal map. Now the major difference you can see between these two is for the NIR, two flight lines are sufficient to create the map, but not so for the thermal. And that is because of the resolution. The near-infrared camera is much higher resolution and higher field of view. So these are the near-infrared maps that we were able to generate. So NIR map 1, 2, and 3. You can clearly see from these maps the fire lines are evolving from map 1 to map 3. And these maps are generated using Agisoft, and we've synced the NIR aerial video and GPS data. So all the maps that you're going to see here from the near-infrared and the thermal are synced with global time and global spatial characteristics as well. And this is the thermal maps. We have map 1 to map 4. One thing you can notice here compared to the near-infrared is the fire line is a lot more prominent on these maps, and I think the thermal band is much more better suited for fire line extraction as well. Additionally, as I said, we had a DJI quadcopter from the Missouri folks that was hovering over the eastern side, and on top is an RGB image of the field. So this had to be georeferenced using an aerial map because we had to remove the tilt and bring it to the same scale as the other thermal maps, and that is the one on the bottom. So as you can see here, the thermal maps, this is the last view of the thermal map that we have, and we had an RGB view of the DJI a few minutes later, and I'll show you guys right now. So now I'm going to show you the fire evolution from the first map till the last map. So this is on an NED frame that is in meters. You can see the time here. So this time corresponds to, so there are two times here. There is the first frame that it sees the fire and the last frame that sees the fire. So I'll go through the entire fire evolution here. So this is a stage where the northern teams are setting the field on fire. So there's a northern boundary here. You can see the fire is evolving. Then you have, this is flight 2. And at this point, the K-Hawk view of the fire ended. So this is the last map we made from the K-Hawk UAS at about 12:17. And we were able to generate another image from the DJI at about 12:21, 4 minutes apart. One thing you can definitely see is the fire line you're having from the DJI is not as continuous as the one in case of the K-Hawk. And that is because the RGB cannot penetrate smoke as well as the thermal band can. But still we have an information of the fire line after at 12:21. So this is kind of the last information we have regarding the fire line. So given this evolution, now we can estimate the rate of spread of fire. So here I'm showing you an example case. That's a plot you see on the top right. Is all the fire lines overlaid on each other. You have map 1, map 2, map 3, and map 4, and DJI. So DJI is basically the last fire line that is yellow. So here I'm going to show you an example case. I'm trying to estimate the rate of spread of the fire at this location. So the way I would do that is I'm looking at map 2 and I'm looking at map 3. So I look at the frame at map 2 that corresponds to this fire line, and I have a timestamp for the frame. Similarly, I look at the frame for map 3 and I have a fire line for that frame, a timestamp for that frame. So given this and given the fact that they're on the same scale, I can estimate the distance and the fire spread speed, which was about 0.4 meter per second here for this field. One thing you guys can see is the fire propagation is significantly faster towards the north than the south, and that is because of the south winds. The winds were pretty strong on that day. So that is using the thermal map. So as you can see, using the thermal maps, we can do fire evolution mapping, and we can also do a fire estimation, rate of spread estimation. Now the near-infrared has an advantage in terms of resolution. So as you can see, even though the near-infrared is able to map the fire, there are still some smoke pixels here. So that is probably better with the thermal cameras where the fire is more prominent. But here, because of high resolution, we are able to identify where the firemen were, the locations of the firemen on the east and the west side. This is again an example case. I'm just showing one map here. So given this, you can estimate where they were, what was the time they were at. And I think this is very good information as a feedback for the fire setting folks as well, for the near-infrared. So I will stop my fire progress there. I'll give you a quick summary before I move on to the next topic. So the K-Hawk UAS was pre-programmed to follow multiple loop patterns for image acquisition, and acquired images were georeferenced, auto-rectified to produce maps, such that we can map the evolution dynamically from across time. And as you saw, the thermal and the NAR maps both have their own characteristics and advantages. Thermal cameras can see through smoke a lot better, more prominent. NAR cameras are have very high resolution. So we can use them for different applications like rate of spread and thermal and fireman location estimation respectively. Now the current and the future directions, what we are currently working on is we want to have real-time fireline detection. So the algorithms that we've developed post-flight, we want to put it on board the aircraft. So the aircraft can make intelligent decisions real-time to follow the fire and predict the fire spread during flight. We also want to work on UAS-based wildland fuel estimation. So given our rate of spread information, and we want to be able to estimate the fuel estimation, the fuel on the field, the biofuels, and maybe we can use it for predictive modeling for future fires. And we also want to evaluate prescribed fire effect on removing invasive plants. So this is kind of our future goal. I think with more data sets and much more flights, we should be able to reach this soon enough. So the next project that I'm going to talk about is the intercomparison of UAS and neon-derived NDVI for hayfield observation. So the overall objectives for this project is now we're trying to estimate the relationships between NDVI from remote sensing platforms with ecological properties measured on the ground, like biomass, for example, at different spatial resolutions. So for this project, we're using two platforms. One is a neon aircraft NIS hyperspectral data, and the other one is our K-Hawk UAS multispectral data. So while estimating the relationship between each of these platforms with ground measurements, we're also trying to understand the advantages and disadvantages of these platforms towards future agriculture applications. Our final goal is we want to develop a data-enabled model using remote sensing to monitor and map the ecological and biodiversity characteristics of grasslands. So for this purpose, we're using a subplot hayfield, a 32 subplot hayfield at KU field station. I think a lot of you may be aware of this field. So this has been maintained since 2000, and Dr. Foster's lab is leading on this front. And the reason we chose this field is because there is significant amount of ground measurements over the years, and I think there's a lot of research and maintenance done on this field that we can use this as a good source to model our relationship between remote sensing and ground measurements. So I'll just give you guys a small recap on the kind of treatments that are going on this field. So there are three main treatments. There is 2 levels of fertilization, 2 levels of seed sowing, and 2 levels of haying. So in total, there are 4 replicates of each treatment combination. So what we're going to do now is we're going to try to compare the NDVI with these combinations and see if we are able to see some trends and see if we're able to extend that further. So a quick talk about the kind of analysis that we've conducted so far in this project. So we started working on this in 2020 in April. But once we started working on this project, I think eventually we realized that there was a lot more work that had to be done with the remote sensing images in itself. So as I said, we're using the images from NIS hyperspectral sensor from the NEON and the K-Hawk multispectral sensor from the UAS. So we observed that hyperspectral data has to be convoluted because those data has much higher, the instrument has much narrower wavelengths and much higher spectral resolution than the multispectral. So if we want to use images from multispectral and hyperspectral sensors together, we have to bring them to the same spectral scale. So we had to perform a convolution there. Another observation we made was the K-Hawk images we acquired were from 2 different cameras. So we had one camera for near-infrared images and one camera for red images. And we saw that there was quite a bit of misalignment and spatial inaccuracies between the 2. So if we want to compute NDVI from this, we have to bring them to the same scale. We have to remove the misalignment. So we've also worked on some co-registration to bring them to the same scale. So once we have the remote sensing data prepared, We've done three preliminary analysis. So first one, we've estimated the correlations between KHOC and NIS NDVI with live biomass for seeded and unseeded plots. So the first one would be, we are dividing the field into seeded, unseeded, and we are seeing how they are relating with each other with NDVI. Next, we are modeling relationship between NDVI and live biomass and combinations of fertilized and hay. So here you're going to have 4 combinations: fertilized, not fertilized, hay, and not hay. We're trying to see if we can see any trends between NDVI and those combinations. And finally, we're also trying to estimate the heterogeneity within each subplot by calculating the variation in NDVI in KHOC and NIS images. So the goal here is, based on the images, we're trying to see if we can quantify the variation in pixels within the subplot, and if that will translate to estimating the diversity of the vegetation within the plot as well. So the first thing was spectral convolution. So here NIS is a hyperspectral sensor, much higher spectral resolution and narrower wavelength than the KHOC. So since the KHOC is a multispectral camera, there is inconsistencies between the 2 spectral resolutions. The image you see is the spectral plot for the KHOC UAS camera, which is a GoPro. So that's your red and NIR, near-infrared. So because of these differences between these 2 sensors, we perform something what you call a weighted sum-based spectral convolution. As you can see in this equation. So this is a transformation that we are performing on the hyperspectral sensor using this information, so that we can match the hyperspectral images with the multispectral images. So this is the first thing, we performed this convolution and brought them to the same scale. And the second one, as I said, was a co-registration problem. So the KHOC, NIR, and red images were acquired using different cameras simultaneously. So if you look at the top figure here before, the left is the red and the right is the near-infrared. The biggest indication that they're not aligned is the road on the northern side here. The road seems tilted on the red image as compared to the near-infrared image. So there are other alignments as issues as well within the field, but this is the most prominent one. So given this, we knew that we had to get them to the same scale. Otherwise, computing NDVI using these 2 would definitely cause some inaccuracies. So we used a point mapping technique in MATLAB, performed projective transformations on this. So basically, we are scaling the red image with the near-infrared image. So we're registering just with respect to this one. And this is the final after image. You can see that the road is straightened out, and they both have a lot more alignment than before. And now we can compute the NDVI for the KHOC as well. So this is the hayfield observed by the KHOC and the NIS images. On the left is the KHOC at 0.1 meter resolution. On the right is the NIS at 1 meter resolution. You can clearly see the difference in resolutions here. And this black box. So what we've done is to extract one value per subplot. So I've drawn a 6x6 window, a box across each subplot. So I take the mean, and the mean of this box will basically correspond to the value of the subplot itself. So in total, we have 32 NDVI values now for each of these platforms. So the first analysis I did was compare the correlations between live biomass and NDVI from both platforms for the seeded and unseeded separately. The left table is the seeded and the right is the unseeded. The biggest indication here you can see, or observation here you can see, is that the live biomass versus NDVI is far less correlated for the seeded plots than the unseeded plots. Here you have about 21 and 6% for the NIS and the KHOC, and for the seeded you have 57 and 43% for the NIS and KHOC here. So this is one observation we made, that this unseeded plots are showing much more correlations between the biomass and the NDVI. The next one we did was analyze the NDVI behavior with different combinations of fertilization and hay. So we have 4 combinations here: fertilized, hay, fertilized, not hay, so on and so forth. So on the x-axis is biomass, on the y-axis is the NDVI. For KHOC, this slide is just about the KHOC UAS. So what we've done is so essentially there are 8 subplots per combination. So in total there are 32. We've done a least squares modeling to generate the linear curve that will relate the 2. We've also computed the correlation R for each of the cases. So the observation we made here was that the third case, where the plots are not fertilized but hayed, showed the highest correlation at 78%, and the plot that were fertilized and hayed, the first case, showed the least correlations for the KHOC. Similar analysis, and we observed similar trends for the NIS as well, where the third case had a 94% and the first case had a 28%. That is the lowest. Another thing is we are noticing here that the NIS neon image is showing much higher correlations with biomass than the K-Hawk. And that is something we are currently working on to see if we can match the K-Hawk and the NIS a lot more. So given those trends, we went ahead and plotted the correlations between the K-Hawk and NIS. So if you look at the plot here, the blues, so these correlations correspond to biomass versus NDVI for each platform, respectively. So here we plotted the blue is the K-Hawk and black is the NIS. So you can see clearly the trends are followed. The minimum correlation is at the green, that is the first case, fertilized and hay, and the maximum is right here, which is not fertilized and hay. So this trend is something we observed, and we think if we can get the K-Hawk to match the NIS more, maybe we can see something better there. So next thing we did was we looked at the coefficient of variation. So what we did for each subplot, as I said, we have a 6x6 window. We calculate the standard deviation, mean, and so we compute the coefficient of variation from both the images. So, and I've plotted them here. You can see the red is the NIS and the blue is the K-Hawk, NDVI at 0.1 meter. So our intention here is we are trying to see if we can observe this variation in pixels between within each subplot, and we can translate that towards diversity in vegetation. Overall, we observed a variation was less than 1% for all the plots. And two things we observed was the maximum was found at subplot 20, where the subplot was fertilized, hayed, and seeded. And the minimum we found was subplot 4, which was fertilized, not hayed, and unseeded. So that concludes our results for so far for this project. So I'll give you a quick summary. So we've done some analysis on the relationship between K-Hawk and NIS and NDVI. And we made some good observations. Number one is unseeded plots are showing significantly higher correlations between live mass and NDVI. And the not fertilized and hayed are showing highest correlations between live mass and NDVI. And finally, we observed that the haying and the seeding have a lot, at least from the image standpoint, have a bigger effect on the variations, because we see the minimum and the maximum values are dependent on that one. So given this, now given that we have the remote sensing images prepared and ready to go, our future directions are going to be that we're going to conduct similar analysis with different metrics like soil pH, PAR, richness, etc. And we also want to perform multiple flights of K-Hawk across the year and also across different years. So we want to map the temporal change in NDVI and ground measurements and see if maybe the NDVI is sensitive enough to capture the change in biomass across seasons and years. Eventually, we want to develop a predictive model. So given NDVI, we want to be able to see if we can map that towards ground measurements like biomass, and maybe estimate the ecological and diversity properties of a given region of interest. And finally, I want to thank everybody who was involved, all the professors who helped out, and specifically the KUFS Conservation Award I got last year. Thank you.
Excellent. Okay, let me just stop the recording here. And